Hi, this is Miss Litton, and this is my wonderful Period 1 AP Biology class. Say hi.
Hi.
And on this Halloween morning, we are continuing our discussion on Chapter 12, Group Notes on Molecular Biology of the Gene, and we are now discussing the structure of DNA. All right? So this is in the 1940s. Chargaff, this scientist, he lived from 1905 to 2002, and they always show these scientists as like old, shriveled up people. And I just wanted to say, look at him when he was younger. Wasn't he handsome? I think it's totally cute, right?
He's a hottie.
So, yes. Like her, Jeremy. Okay. So, anyway. Okay, thank you. I was looking to see what you're doing. Okay. So I just thought I would show you what he looked like. Now, several scientists, there are books written, one in particular, about the chase for understanding, not Martha Chase, but the chase for understanding the structure of DNA and understanding our blueprint, our code book for life. So Chargaff didn't come up with it, but he had evidence that contributed to Watson and Crick's understanding of that. And that was about the bases. And when he analyzed different species' DNA, he found that the amount of adenines was about the same as the amount of thymines, and the amount of guanines was the same as cytosines. Now we know DNA to be a double helix, a twisted ladder, right? They had no idea the arrangement of it. So this is a big deal. So the idea that we now know that there were pairings between them, this still had to be discovered yet. He knew that however many adenines, thymines, and guanines and cytosines you had were specific to your particular species, but at least the A's and the T's were about the same, and the G's and the C's were about the same. So if you have 4 bases to work with, you could remember our earlier discussions consisted of could we have a protein which had 20 amino acids versus DNA which has 4 bases. These bases can be repeated in any pattern, and since the human chromosome contains approximately how many base pairs? 140 million base pairs. Any of the 4 nucleotides can take up any one of those spots. So it does, even though there are 4 bases, lend itself to enough variety. Yes?
When this guy was doing the experiments, did he also find that like the A and the adenine and thymine, like was that the same amount in each, or just?
That will vary between the different species, but the amount of A equal here. I'll show you what he came up with. See, they always throw him with now the big eyebrows and the old man look. But these are his two rules that he came up with. That the composition of DNA would vary from species to another. So each species has a certain number approximately, like humans have a certain number. We're kind of almost even on ours. But in any DNA, the number of A's was the same as the number of T's, and the number of G's and C's were about the same. That's what he came up with, okay? So if you take a look at this chart, Blue is going to explain to you what this chart means, and they'll have no trouble doing that. Go ahead, Blue. Look at the chart. Now, is it exactly the same? No, but it's pretty much the same right in there, okay? And we have more A's and T's than we do what? C's and G's, okay? So when you look at the human DNA composition, it looks something like this. So there were some other scientists as well, Rosalind Franklin and Maurice Wilkins. And what they were looking at was X-ray diffraction studies, which basically they took crystallized DNA and sent some X-rays through them, and they went and they had this pattern. This is a classic photo here on the right. Classic. I'm not kidding you. It shows up on tests, and people who study X-ray diffraction, they go, oh, look at that. Look what I can tell. Now I want to show you something so that you can tell what this would be. Have you ever, a spiral staircase? So imagine you're at the top of a spiral staircase and looking down through it. Okay, that's what you're seeing here, like the steps spiraling down. So they could see that. There was some repeating patterns, it was the same diameter, and there was some spirals. Now, this was a really big deal about Rosalind Franklin and Maurice Wilkins. They were trying to keep this information to themselves because everybody was racing to come up with the structure. And so there was some sneaking around going on that Watson and Crick got this information, and that's all debatable. But between Chargaff and Rosalind Franklin and Maurice Wilkins, they were able then to come up with the structure of DNA in 1953. Now, your parents were probably born in the 60s, maybe. I don't know if you know when your parents, but your grandparents were alive during this time. Would you agree? So it's not like it's that long ago. Now we're at the point where we're manipulating and cutting and splicing DNA and genetically engineering organisms and making clones, you know, and it wasn't that long ago that we even figured out the structure of DNA. They also had bond angles. Remember how we talked about oxygen being a polar molecule and how it'll tightly hang on to the electrons, and so they knew bond angles as well, Watson and Crick, and all of that came in to play when they came up with the structure of DNA. Now, if you look up, let's see if I can get you here. Yeah, you can. I don't think you can see it very well, but you do have put a picture of DNA definitely in your group shared notes. You can see that twisted helix, and it is 2 nanometers in diameter, and I don't think you can see this. There you go. So it is 2 nanometers in diameter. The space between each rung of the ladder is 0.34 nanometers, and a complete turn, like if I'm facing this and I'm going for a complete turn, okay, it's 3.4 nanometers, which means that you have 10 base pairs in a complete turn. Okay, you can see the hydrogen bonds that are holding. How many hydrogen bonds between adenine and thymine? 2. How many between guanine and cytosine? 3. Okay, so that, and here's a picture of Rosalind Franklin down there. So let's do a little review because I can tell that you are not quite getting that. Well, look, my little thing's not coming up. Oh, there it is. Okay. So here is a nucleotide, yes? Base, sugar, phosphate. Is that a purine or a pyrimidine? It is a pyrimidine because it only has 1 ring. That is cytosine. Who would he bind to? Guanine. Okay, boys and girls, we need to review this a little bit. There are 2 kinds of sugars, ribose and deoxyribose. Which one would you find in DNA? Deoxyribose. Okay, now I want you to pay attention, and we talked about this before. Look at ribose and look at deoxyribose. Okay, there's a hydrogen right here. What is it missing? Oxygen, deoxyribose. Now what I want you to pay attention to is right here. These are your carbons. This is referred to as carbon number 1. You can, and we can't go, if this is a house, we can't go on the roof because what's on the roof of the house? What is right there? That's not a carbon, that's a what? Oxygen. So now we come down, here's carbon number 2, 3, 4, and the chimney is number 5. Do you see that? It would not matter if this house was right side up or upside down, we could number the carbons all the way around. Okay? So when we look here, okay, we can see this is a single nucleotide that I've highlighted right here. And DNA is 2 strands of nucleotides held together by hydrogen bonds. But I want you to pay attention to the sugars on either side. And youngest bio buddy, what do you see when you look at the sugars on either side? Do you see how the houses look right side up and then upside down? Do you see that? Okay. So now watch when I point this out to you. I want the one on the left. Okay. Here's thymine. Where that bond is, what kind of bond is that, by the way? It is not a hydrogen bond. Hydrogen bonds are here, the dashed ones that hold the 2 strands of nucleotides together. What bond would this be right here? A covalent bond. Carbon number 1 is the carbon that's attached to the nitrogenous base. Carbon number 1 on the sugar. So here's carbon 1. I told you what's at the top, what's at the roof that we can't count. Oxygen, so we have to go down. What's this one? 2, 3, 4, chimney makes 5, right? Go across the street over here. Here's the nitrogenous base. This is carbon number 1. Okay, I'm not going to go down here because that's oxygen, right? I'm going to go up. 2, what? 3, 4, chimney makes 5. So when you look at DNA, you can see up at the very top, faintly, it says 5' in, 3' in, 5', 3'. Do you see that? And that's because when I look here, okay, here's the base, there's carbon, that would be carbon 1, 2. This one right here is number 3. That is why it's called the 3' end. What do we know about proteins? They are always very what? Specific. And enzymes are very what? Specific. Enzymes to copy DNA, to make more DNA. By the way, when does that happen? What stage? S stage of what? Interphase. When you go to replicate your DNA, the enzymes can only read 3 to 5. They cannot read 5 to 3. So we're going to talk about what's the workaround for that on how enzymes work. It could read this 3' end right here. If the eyeball is looking, I'm looking at a 3' end right here. But if I go to this other end, here's 1, 2, 3, 4, and right there is the 5' end. So DNA could be copied going this way, but it would have a hard time going the other direction if you're trying to copy the DNA. If we go right across here to the other side, this is the 5' end. Can I copy that? This is a 3' end. Can I copy that? But what happens if I'm starting right here? And I'll tell you what they have to do. The enzyme has to wait for some of the DNA to be exposed and go back a little bit, turn around, and work its way back this way, and work its way back this way. And that's why you have a lagging strand in DNA replication. Now, I'm going to explain more of that in a minute. I'm just trying to set your brain up for it by looking at that. And I would like, please, the youngest bio buddy to explain what you see here about numbering, etc. Go. All right, now I want to fill in the notes. So, structure of DNA, Chargaff in the 1940s. Let's go back and visit Chargaff. You have analyzed DNA-based content but did not know its connection to structure. B, DNA was found to have the constancy required of the genetic material constant to the species, even though only 4 options. A DNA molecule of a human chromosome has about 140 million base pairs. They can provide enough variability. Chargaff's rules. The amount of A, T, G, and C varies between species. However, in each species, the percentage of A's is going to equal the percentage of what? T's, and G's is going to equal C's. Good. Rosalind Franklin produced X-ray diffraction photographs. X-ray diffraction photographs, and showed that DNA is a helix, constant diameter, and has repeating portions. And when you look at the structure, if you can, I mean, maybe you'll never use this, but if they give you an essay on DNA, DNA is number 1, right? It's our code book. You just got to remember 2, 3, 4. 2, you remember the number 2, because there's 2 nanometers. When you look at DNA, the width right here is 2 nanometers. Okay? Then the next one you have to remember is 0.34. And that is because when you look at the base pairs, there in between each of these base pairs is 0.34 nanometers. and then just move the decimal one more time, 3.4, that's how much one turn is. Okay, is 3.4 nanometers. Just remember, DNA is number 1, 2, 3, 4. Diameter, bases, and a turn. Okay? Diameter, bases, and a turn. Okay, and then, so a helix, constant diameter, and repeating portions. Then Watson and Crick, 1953, worked out the structure of DNA using Chargaff and Franklin's data. They received the Nobel Prize. The structure is a double helix with a sugar phosphate backbone, right? Alternating sugars and phosphates, which we can see right here. Right? Sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate backbone. And paired bases, a purine with a pyrimidine. Why did I write that last part of that sentence? A2HT, G3HC. Number of hydrogen bonds. Okay? You ready for some questions?
I can't remember the number. Is it
You can't get in?
Or just 3-1?
Oh, it's 314.
314 number. 8679. 8679.
Oh, 8675.
530. Yeah.
I got Legend Week for Dairy. I got the best. I got lucky. I got lucky. I got lucky.
314.
That's what it is.
I know it's stupid, but
Do you have a cool shirt on?
Yeah.
Oh, no. Yes, you do.
Oh, my avocado.
Nice.
Oh, yeah.
What does it say on your avocado shirt?
I don't know, avocado. There's an avocado
It's an Israeli company.
Oh.
They're doing excess of avocado.
No, there's a liquid like some places aren't getting avocados.
Oh, no.
Hashtag stay updated, Max.
All right, that's it. That's all the time I'm giving you. No. Okay, phosphorus, base, sugar, what? Phosphate. So phosphorus was located inside the bacterial cell as part of the transforming agent. Show your clicker to your bio buddy. Gong of victory
Gone.
or a shame bell? Are you ringing a shame bell? Bell of shame.
I got
Thank you. Still haven't gone. This isn't the easiest one to say.
Oh, good job, Puzzles, Lesbian Robin, and the Mermaid Effect.
Puzzle.
Okay, what did they say? Why do we not like B? The number of, yeah, what do A and G have in common? They're both purines, but your number of adenines is not going to equal the number of your guanines. Okay, they're just both purines. Look at your bio buddy's clicker. Chastisement, praisement, bell of shame, or a gong. Just give me a number. You don't need to type in percentages. Let's go, peeps. Focus. My Halloween children. Stop. Are they new?
Wow.
clap that. Oh, good Lord, no.
30.
Well, you know what they were thinking. Yeah. So 20% of the bases were thymine. Who does thymine bind with? Yeah, he binds with adenine. So if 20% of those bases are going to be thymine, then the other 20% have to be what? Adenine. Because matchy-matchy. Check with your bio buddy. Gong or shang? Gong or shang? Oh.
I'm never number one.
Let's go. Okay, Pete. Guys, I need you to dial in, okay? Because when you don't understand, and I've spent a lot of time explaining, then it slows us down, okay? So 20% of the bases were thymine. So T is 20%. Who does thymine bind with? Adenine. Okay, with adenine. So it would also be what? 20%. What is 20 plus 20? 40. What is left then? What percentage? 60. How could we divide that up?
By 2.
Yeah, if we do 30% and 30%, it'll all work together. Check your bio buddy's clicker. Chastisement, praisement. Good job, lesbian Robin. Let's go. This can be your hundy. You can do it. I believe in you. Yeah, I don't want to do it. You do it. You know what?
No, I think it's dry cleaner.
Well, I didn't want to do that.
School sweaters. School sweaters
Okay, guys, focus back because we need to still help somebody. 15% of the bases are cytosine. Who does cytosine bind with? Guanine. And how much will be guanine then?
15.
And that equals?
30.
30. So what is left?
70.
How can we divide that in half?
35.
Is that right?
Yes.
And that would be adenine and what? Thymine. Okay, check with bio buddy. Check for understanding because somebody's still struggling. All right, take a look at this diagram. My friends, your large mammalian brain can label everything on this diagram right now. Those who are the flip-a-pics, you may want to grab this before and grab it after. You should be able to label this diagram. Talk about, please, quietly and quickly with your bio buddy. How would you go about labeling this? What is what? And then Also, I'm on 3 and 5 prime end labels. Okay, tell me, where do you want to start? The bonds, okay. So where am I going to put this? Right there, and 2. Are we good? Okay, and we could do that for all of them, but you got it. Okay, what else? Phosphates. Where are my phosphates? What shapes? Circles. Excellent. Okay, that's good. I've done a few. Okay, what else did I do? Deoxyribose. What is that? All the houses. Okay, good. Let's label now, please, how we know 1, 2, 3, 4, 5. What are we going to do here? What is this one going to be right here? That's going to be 1, 2. Because why aren't I going up here? Oxygen. And this is the 3. So this has got to be the 3' end, and this has got to be the 5'. It's one of those. This is hypertonic. This is hypotonic, right? Plus you could figure it out, right? So here's 1, 2, 3, 4, 5. Okay? So then let's go over here. Here's where it binds. I can't go here, right? So it's 1, 2, 3, 4, 5. 5' in, and this is the 3'. And I can check because here's 1, 2, 3. We all right with that? What else can I label? Okay, let's do the adenine, and who does adenine always bind with? Adenine and thymine. I'm not putting them anywhere, I'm just pulling out the labels. Okay, how many hydrogen bonds between adenine and thymine? 2. So I need to go to some Dublin, right? And how do I, here's one that has 2 hydrogen bonds up on top. How do I know which one's adenine and which one's thymine? Adenine would be the bigger one because it's a what? Purine. And this would be thymine, the pyrimidine. Okay, now let's tackle our next one. What's the one on the left? Guanine, because it's the bigger one, and it always binds with cytosine. Do you see how smart you are?
Yes.
Okay, you are super smart. Look at that. I would bet most people in the world couldn't see a naked ladder of DNA and go, let me tell you what everything is.
That's right.
Now, I told you already before, when we replicate DNA, right, there's going to be what's called a leading strand and a lagging strand. The leading strand is the one we're going to be able to use when it starts with a 3' end. If it's a 5' end, it can't read the sugars. You can see the sugars are going the other way. That will be our lagging strand. So when I point to it, you tell me leading or lagging. Leading, because it's 3'. Good. So when you start somewhere in the middle of this, if you're in the middle of the DNA, I'm right in the middle of the DNA. If this is leading, right across the street is lagging, then over here, if this is leading, this one must be lagging, and this one must be. So you can take off on 2 of the ends super fast, right? Both of the leading parts, you can take off really fast. But on the lagging parts, that's the slower one. That's where you have to travel a little bit down the road, turn around, and come back, because you can only move in one direction. Okay, and that's going to be our next topic, but I'm going to stop this right now, and then we'll pick up, because you like it when I break it up, yeah? DNA replication. I hope you're having a good day, and if it's late at night, eat some toast. Oh, and happy Halloween.